All around you, trillions and trillions of bacteria, viruses, and other pathogens roam freely. Without any kind of defense, your body wouldn't be able to survive, so over billions of years, animals evolved the modern immune system. The immune system can be generally split into three distinct categories: our first, second, and third levels of defense. Our first layer of defense is quite simple. It's just a physical barrier, preventing pathogens from entering our body in the first place. This includes the skin, stomach acid, tears, sebum, etc. So let's say a pathogen makes its way past our first layer of defense. Instead of preventing microbes from entering our body, our second layer of defense actually kills pathogens, just very generally. Macrophages eat all kinds of invaders. Lymph nodes and lymph vessels also clean bodily fluid. Complement proteins, along with inflammation, are also parts of the second line of defense. What's important is that the second line of defense acts non-specifically. The third line of defense, however, is specific, tailored to each pathogen individually. It takes time to activate and set into full motion, but is extremely powerful. To start, how does the body even distinguish between cells belonging to the body and invader cells? Well, every cell has proteins on its surface known as antigens, which act as identifiers, a kind of cellular name tag. These antigens are key components in recognizing all other cells. White blood cells react to different antigens for different purposes. Following the immune responses from the second line of defense, a new cell enters the playing field: dendritic cells, another type of white blood cell that has long tentacles called dendrites, hence the name. These cells use their long arms to collect remaining antigen fragments belonging to the invaders from the point of infection. Think of these cells as detectives that take evidence from the crime scene and bring them to the FBI. Dendritic cells present antigen fragments in a protein called MHC class II, but more on this protein later. Dendritic cells take advantage of our body's complex transportation system, the lymphatic system. This system is comprised of lymph vessels, which are like superhighways that carry cells quickly throughout your entire body, along with the lymph nodes, which act like cities where vital white blood cells may stay reserved in the body, ready to act at a moment's notice. The dendritic cells enter the lymphatic vessels and make their way to the lymph nodes, which contain large groups of T cells. T cells are a type of white blood cell that play a central role in your specific immune defense. There are two subsets of T cells that are important to remember: helper and killer T cells. Helper T cells are specially tasked with activating other types of white blood cells that are critical to the adaptive immune response. Killer T cells, also known as cytotoxic T cells, are granted the ability to kill cells that may have been infected by a pathogen. When the dendritic cells finally arrive in the lymph nodes, they need to search for the exact T cell that recognizes the antigen that are presented to them. Your thymus trains millions of T cells that are all specialized to recognize individual invading pathogens. Actually, something really cool about T cells is that there's a T cell for any possible antigen. Any pathogen that has ever existed, that ever could exist, there's a T cell for every single one in your body. One for the bubonic plague, one for SARS-CoV-2, and so on. When that special, unique T cell is discovered, the dendritic cell must activate it by presenting fragments of pathogenic antigens to the inactive or naive T cells. A process that takes around 2 days in order to allow the second line of defense to do what it can and to prevent unnecessary inflammation, which may result in the vital loss of resources. These pathogen fragments are presented through proteins known as major histocompatibility complexes, or MHC for short. There are two different classes of MHC that are used to activate different cells at different times. The first, MHC class I, is present on almost every type of cell in your body, and it acts as a window into the cell. Cells present fragments of the proteins within through this window, and other cells can identify the status of these cells to discover if they are infected or not. The other MHC class II is only present on the professional antigen-presenting cells like dendritic cells or macrophages that have the job of alerting T cells to infections within the body. This MHC is more like a hot dog bun. It holds small antigen fragments from bacteria or viruses, think of these like the hot dogs, within the buns, and can then give those hot dogs to other T cells to activate them. As a note, T cells can only be activated when recognizing an antigen fragment presented in an MHC class II protein. After the T cell is activated, it multiplies itself, making more and more copies of cells that can help protect from the active threat of the intruder. After both killer T cells and helper T cells undergo this process of multiplication, they can assist the body in their own ways. The killer T cells initiate what is called a cell-mediated response. This response is useful for targeting pathogens like viruses that may overtake body cells as hosts and then removing them forcefully and violently. First, killer T cells are activated by MHC class II on dendritic cells, just like helper T cells. They're sent to the site of infection where they recognize that a cell has been infected by looking through the windows of the MHC class I proteins. When activated by an infected cell here, they kill said infected cell by releasing harmful chemical packages called granules, which contain perforin and other cytotoxin, agents which are dangerous to cells. When these granules are released, the cytotoxins within begin to act in waves against infected cells. The perforin pokes holes into the cell membrane, rupturing it in a process called cell lysis. This lets the other cytotoxins enter the cell, inducing apoptosis, a programmed cell death that eliminates cells that could be harmful to the body. The helper T cells, on the other hand, have another job. They need to go activate the other fundamental cells that make up the immune system's adaptive response. They help to maximize the bactericidal or bacteria-killing powers of the phagocytes or eating cells like macrophages. They also need to help activate the naive or memory B cells. B cells are especially important and begin activating the humoral response, a response in which the aim is to kill the pathogen before it can enter the body's cells. Just like T cells, there exists B cells for every possible antigen. B cells mostly float around in lymph nodes. When intact antigens from the site of infection float through the lymphatic vessels, virgin B cells stand in the way, letting lymph flow around them until one of them binds with an intact antigen. This activates the virgin B cell where it will begin multiplying and producing low-grade antibodies. These antibodies are specific to the antigen, but aren't especially great at their job. These B cells break down their antigens into antigen fragments that helper T cells can recognize. When these two cells meet, this activates the B cell and it specializes into a plasma B cell. Plasma B cells are antibody-producing machines, creating antibodies at an insane rate of about 2,000 every single second. Plasma B cells also do something special. They purposefully mutate their genetic code to create slightly different receptors, and hence antigens because they're created from the same genes, which will simulate natural selection to create a plasma B cell with the perfect antigen. Antibodies created by plasma B cells clump pathogens together and debilitate them, killing the infection. After an infection is dealt with, many immune cells are ordered to commit apoptosis as they are no longer needed. However, some immune cells advance into memory cells. Memory B cells remember the antigen that activated the original infection and will float around in the bloodstream. When they are re-exposed to the antigen, they will divide into plasma B cells and restart antibody production. This is why severe infection is unlikely if you are reinfected by a pathogen that has already infected you. Memory T cells can recognize their antigen well and immediately differentiate into killer T cells, allowing for faster response. Memory cells help prevent reinfection. As an example, if you get a vaccine for a pathogen, many vaccines work by either injecting antigens or fragments of the pathogen, which are on their own harmless, but cause an immune response to create memory cells, which will recognize future infections of said pathogen. Here is an overall look at the third layer of the immune response. We see the chain of events that occurs in the humoral response, from helper T cells to B cells to plasma B cells, and in the cell-mediated response, how the killer T cells are activated and go through with their job of killing infected cells. It's important to remember that the cell-mediated response acts on pathogens that have already entered and infected cells, whereas the humoral response focuses on pathogens that are free-floating throughout your body.